So Jorn is actually one of the leaders in motor control using fMRI, and he's especially focused on cerebellum. He has his PhD in UC Berkeley, and then he joined Johns Hopkins. This is the time when we overlapped in Reza Shadmer's lab. And later on, he became a faculty in UCL, and now he's been a faculty in Western University, I guess, for a few years. He is one of the highest cited, I would say, motor science neuroscientists, and both in terms of modeling work, in terms of the psychophysics, developing toolboxes for fMRI. And so it's a great pleasure to have him here to share with us some of the recent development and advances, both in terms of science and methods and whatever he finds basically worthwhile to tell us. With that, Jorn, the floor is yours.
Thank you, Alex. Ali, it's great to see you again after such a long time.
Yeah.
Good. Yeah, so let me share my screen and then see if this works here.
So if you click on the...
Yep.
Okay,
Is that, can you see my screen?
Yes, we can see the screen.
Okay, so let's see. Is that good?
It is good now.
Yeah. Okay. I can't see you, unfortunately, in this platform. But what I'm gonna do is the talk has 3 parts, and after each part, I'm gonna just stop real quick and see if there are any questions. So please do feel free to ask questions in between. It makes it easier for me to pace myself through an online talk. Great. Good. And if there are questions in between, just shout, because I can't see the chat or anything. Great. Well, it's a pleasure to be, at least virtually, with you guys. Today I want to talk about one of my favorite brain structures, namely the cerebellum, the human cerebellum, and especially I'm going to talk about the function of the cerebellum, not only in motor control, but across many cognitive domains. Okay, and this is work that we've done over the last 5 to 6 years or so that our lab really got into and interested in. Now, I like to start this talk with this image here. This is a MRI scan of a human cerebellum that we took from a postmortem cerebellum at 150 micron resolution. And I'm just showing this because it shows really how incredibly folded the human cerebellum is. Okay, so you see here at 150 micron resolution, you can see the granular cell layer and the molecular layer of the human cerebellum, and this incredibly folding of these folia that are very tightly packed in this magnificent structure. Now, based on the scan, Marty Serino and I, back at UCL, used this data to now provide actually the first full surface reconstructions of a human cerebellum. So this is the reconstructed surface with green, the top of the folia, and red, kind of the bottom of the folia. And you can blow it up, and then you can try to spread the human cerebellum out, and what you get is a sheet that's about 15 centimeters wide and about 1.2 meters long. Okay, we found that it has about 80% of the area of the entire neocortex, so it's actually bigger than one of the cortical hemispheres. You know from other data that the human cerebellum has more than half the neurons of the human brain, and that's mostly due to these granular cells, from which we have so many. It uses about 1/5 of the total brain metabolism, although it's only about 1/7 of the volume, so it's quite an energy expensive structure. And remarkably, and this has become clear over the last 10, 15 years, not only in mammalian and primate evolution, but especially in human evolution, it has expanded more than the neocortex. So the recent data shows that from the Neanderthal to Homo sapiens, the biggest difference is the size of our cerebellum. So from this, you can infer that the cerebellum is extremely important to make us the humans that we are. But the key question, and this is a question that I want to kind of circle around in my talk today, is what does the structure actually do? Now, you know, if you look at textbooks, the answer is simple. The human cerebellum is there for motor coordination. Okay, it makes your movement smooth and coordinated. And this is based on the fact that the cerebellum is connected to primary motor cortex. Now, the story became much more complicated in the mid-80s when people realized that it's not just the motor cortex that connects to the cerebellum, but basically every other cortical structure as well. So here, this is in this cartoon, these are these cortical areas that have all some interconnection, and they all project to different areas of the cerebellum. Okay, and so Leiner and Dow in mid-80s suggested, based on this data, that the cerebellum isn't only important for motor control, but also important for cognition. And this really constituted a watershed moment in cerebellar research, where the question became, well, what is it that the cerebellum does? Now, since then, we know much more about the connectivity of cortex and cerebellum. And for example, Kelly and Strick with advanced viral tracing techniques showed that the cerebellum seems to engage with the cortex in these closed loops. So they showed that primary motor cortex projects to lobulus 5 in the anterior cerebellum, which then projects back exactly to the same area. And they also found that dorsolateral prefrontal cortex, DLPFC, projects to an area called CRUISE2, and this area also projects back to this area, DLPFC. Okay, so based on this data, they suggested that how the cerebellum works together with the cortex is in form of these closed loops, where each cortical region projects to the cerebellum and the cerebellum region projects back. I should say here that there's very little interconnectivity between different cerebellar regions. Okay, so it's really in and out circuit. Now, at the same time, from human fMRI, what we've learned is that the cerebellum, basically human cerebellum, is activated for all kinds of tasks, not only motor tasks, but for working memory tasks, language tasks, social cognition, what have you. Whatever activates the cortex also activates the cerebellum. Now, when you look at the cerebellum, this is one of the circuits in the brain where we have actually for a long time now, for more than 50 years, a very, very good understanding of the underlying circuit. So just to review this, and most of you should be familiar with this, the human cerebellum has these loops with neocortex, and the neocortex projects about 40 million fibers down into the brainstem. And from which 90% actually synapse in the pontine nuclei, at least with collaterals. Okay? The pontine nuclei then give rise to these mossy fibers that synapse on a whooping 50 billion granular cells. So this is a more than 1,000 magnification of information. Okay? Each granular cell listens to 4 mossy fibers and gives rise to this massive parallel fiber system that then is being listened to by the major output cell of the cerebellum, the Purkinje cell, from which we have about 15 million. Each Purkinje cell gets about 175,000 of these parallel fibers. Okay, and the Purkinje cell now inhibits the deep cerebellar nuclei that then project back to the thalamus and back to the cortex in this closed loop. And then we have, of course, inhibitory interneurons that complete the circuit. So it's really this in and out circuit with relatively little lateral connections and little internal circuit. Now, an important input to the cerebellum is the inferior olive that gives rise to climbing fiber, and every Purkinje cell basically gets one and only one climbing fiber. And the learning rule that underlies the cerebellum is also well known and in broad strokes was already published and suggested 50 years ago. It's the Ma-Albers-Ito theory of cerebellar learning, that basically what Purkinje cells do is they try to anticipate when a climbing fiber input comes by looking at this parallel fiber in the inhibitory interneurons and trying to learn to anticipate this, and then in anticipation actually lower their firing rate to give a signal back to the cortex. So there, as Ma puts it in his 96 paper, it's a perfect predictive device, if you will. Okay, so we have all the pieces in a way. We know the overall connectivity with the cortex. We know what areas they are activated for, and we have the circuitry. So could we figure out what the function of the cerebellum is? Now, the main idea is that the circuitry that I showed you is basically preserved across all these areas in the cerebellum. It's not like the cortex where you have different Brodmann areas. The cerebellum is really homogeneous in its circuit across all these different circuits. Okay, and so this has given very early on rise to the idea that also the function should be the same, meaning the way that this cerebellar area helps primary motor cortex to keep your movement smooth and coordinated, it would be the same way that cruise 2 helps dorsolateral prefrontal cortex to keep your thoughts smooth and coordinated. Okay, and this idea, has been suggested by multiple people, but has been then formalized into the word universal cerebellar transform by Schmarman, who coined this term. The key question is, of course, what is this computational function that the cerebellum really contributes? And there have been, of course, many suggestions. For example, Rich Ivory, you pushed the idea together with Thompson that it's timing. There's like it learns some time, exact timing. Chris Meyer and Daniel Wolpert suggest the idea of forward modeling, and so on. There's a number of suggestions. Now, I think the sobering thing that we need to admit, even after 20 years of research in this area, that we have unfortunately made very little progress to test and really falsify these different ideas. So I don't think we have a full answer of a full explanation of how the cerebellum works in these different circuits. And in my talk today, what I would like to do is to highlight some of the reasons why that is, but also try to see how we can use human fMRI to maybe make some progress into this question. Now, with human fMRI, it's good to know, first of all, when you use it on cerebellar circuits, what the strengths and what the weaknesses of the techniques are that you're using here. Okay. Now the strengths are, of course, that you can see activity in the entire neocortex and the entire cerebellum of a live human being at the same time. So this is pretty cool, obviously. But what are the limitations? Well, the limitations are, first of all, that the signal that we don't see neural activity directly. We can't see spiking activity. What we're seeing is the hemodynamic signal, namely the blood oxygen level dependent signal. And that depends on how much blood actually rushes or is supplied to a specific area and how much oxygen is being taken up. So to understand what the blood flow in the cerebellum means, we need to understand how blood flow to the cerebellum is regulated. Now, it is true that in many brain areas, we have actually very little idea about what exactly leads to blood inflow. But in the cerebellum, we are actually in the lucky situation that there's a Danish group across around Martin Lauritsen that over a decade has published a whole series of papers looking at blood flow regulation in the mouse cerebellum. And what they find basically is that the blood flow is regulated mostly by these stellate cells that emit a nitric oxide emission signal that then leads to blood flow increases. And that would lead then to increase in Bolt signal. The stellate cells get input from parallel fibers, which get input from granular cells, which get input from mossy fibers. Okay? So when you stimulate pontine nuclei, you stimulate mossy fibers, you stimulate granular cells, or you stimulate stellate cells, you get increases in blood flow. Now, the interesting thing is that this major output cell, the Purkinje cell, you can actually make them fire twice as high by shunting off the inhibition here, and you don't see any changes in the blood flow. So basically, the Purkinje cells are blind, or the fMRI signal is completely blind to Purkinje cell firing. At least that's what it looks like from this data here. So this means that we actually, when we look at cerebellar cortical Bolt signal, we need to admit that we're mainly looking at the input and how inputs arrive. And we don't actually see the function of the main computational unit of the Purkinje cell. Okay, and that's certainly one limitation in fMRI that we need to take into account. Now, the other limitation is that of spatial resolution. As I showed you, the folia are very tightly packed, much more tightly packed than the cortical folding. And even with a voxel resolution of 1.5 millimeter, which is pretty high, you're not going to be able to resolve these different folia. Okay, so you're going to average around it. Now, based on this limitation, we have early on, back then in Reza's lab even, I started this to develop an atlas template for the human cerebellum that looks like this, that is now kind of quite standard in the field. And we also developed a fake flat map that just is basically the hull of the cerebellum where you can project and view activity patterns on. So this is kind of the hull, and then you can kind of flatten this out, and then you can see the activity patterns on it. Now, the way that people think about different cerebellar regions is in the terms of the macroanatomical folding. And this notation was first proposed by Larsal, and then for the human cerebellum formalized by Schmarman et al. And it's in terms of these lobuli. So lobula 1 to 3 are very small, 4, 5, 6. So they're denoted with Roman numerals. Lobulus 7 is the largest one. It consists out of crus 1, crus 2, and 7b. So this accounts for about 50% of the human cerebellum. Yeah. Then you have lobulus 8, lobulus 9, and then the nodulus and the flocculus and lobulus 10. Okay. And this is how people review and talk about location in the cerebellum. So in many imaging experiments, you will see this, but also for neurophysiological recording. All right, good. So now we come to the main part of the talk. So the talk has 3 parts. And in the first part, I'm asking the question, well, what are actually, if these different regions talk to different regions in the cortex, what are actually the main functional regions in the cerebellum? Okay, we have some idea for meta-analysis, what activates what, but what is actually good postulation, or how should we think about the cerebellum as being made up in the human? And then in the second part of the talk, in third part of the talk, I'm going to talk about how these areas connect to neocortex and what we can learn about this for the function. So the first question, what are the functional regions of the cerebellum? Now, when we started, there's meta-analytic approaches, but also approaches, a few multitask approaches. But really what was missing was really a comprehensive data set where you could see in single subjects across many, many, many different tasks, what is actually the cerebellar activation pattern looking like? Which parts of the cerebellum and how are they arranged? Okay, and so this is a task that Maeve King, who was a master's student in my lab back then, and Carlos Hernandez, who has now his own lab at Dalhousie University in computer science, started. And basically what we did was we went across the cognitive neuroscience literature and picked many, many different tasks, from motor tasks, theory of mind, action observation, what have you, working memory. And we basically randomly sampled these different tasks. Okay, and we generated 2 task set, one with 29 task conditions, one with 32 task conditions. We have 14 of the task conditions were shared. And the experiment was basically, we put people in the scanner, 5 seconds of instruction, 30 seconds of task, one of these tasks. Many tasks had multiple conditions here. And then another 5 seconds of instruction, another 30 seconds of task, and so on. And that basically for 8 hours total per subject. Now, what I'm showing you here on the flat map are some of the example activity patterns that we get. So what you get is basically what you expect as a motor control person. You can kind of see here for finger sequences, for example, you see the activity in the superior and inferior part of the human cerebellum. These are the motor regions of the cerebellum, okay? And you also see that during simple finger movements. But what was surprising to me as a motor control person, although I knew from other literature that this would be the case, that here are some tasks that do not have any movement at all. So this is a theory of mind task where you just are supposed to read some stories about other people and think about it, like Harry loves Sally or not. And what you see is this very strong activity in these very replicable activity cross experiment in this mid-medial regions in crus 1 and crus 2. Watching other people tying knots, so this is also no movement, you just have to observe people how they tie knots, leads to very different activity patterns here, basically surrounding these motor regions. So this is action observation. Okay, verb generation, this is like when you read the word dog and you have to think of a verb that goes along with, like bark, that is this task here. It leads to this very right lateralized language network. If in case you've forgotten, the right cerebellum connects to the left hemisphere, so that's why all the laterality is flipped. Okay, and you also get this resting state network here. So if you're interested, there are many, many more tasks. If you're interested, if you have your own data, if you want to know what does a specific area of the cerebellum do, you can go to this web address here. So it's an image viewer that a computer science graduate student of mine programmed, where you can actually view these different activity patterns, both in the volume and on the surface. And it's kind of really nice to explore this. It also has data from many other people in there. Now, how do we summarize these data? So we have these nearly 60 tasks or so. A good summary, maybe we can achieve that with clustering. That's what we did. So we took all these contrasts and we used clustering. In this case, a semi-non-negative matrix factorization. It's kind of a form of dictionary learning. This is the parcellation that we get. Okay, here in dotted line, you see the lobular boundaries. This is how people normally parcellate the cerebellum. And here is our functional parcellation. And you can kind of see this network of regions here that goes from medial to lateral in Cruz 2 and has these kind of funny shapes. It's interesting that previously people have done similar parcellations using functional resting state data, so where people just lie at rest in the scanner and you look at the normal fluctuations, and they actually do look stunningly similar. Actually, they have quite some overlap with this one. Now, the advantage of doing a task-based parcellation is, of course, that you can understand what the function of each of them is. So we could give them functional labels. And to do this, what we did was we went across our 60 tasks, and together with Russ Poldrack at Stanford, who has worked on what's called the cognitive ontology, we kind of rated those tasks on different cognitive terms, so theoretical terms that cognitive scientists use to describe different task contrasts. Okay, and we rated them. We rated the task on this. We also put in, for example, the actual measured number of left-hand responses, number of right-hand responses, and number of eye movements. And so when you do this, what you find is that that this area and this area are, of course, connected to left and right-hand movements. That's the term that sticks out in a regression analysis. You also have the lower part here. There is the so-called ocular motor vermis that is highly explained by the number, just by the number of saccades across tasks, how many saccades you have to make, and how many saccades you have to make. There's this action observation network that every time biological motion is on the screen, either in movies or on the action observation task, is highly active. You get these working memory networks, both on the left and on the right side. They have a little bit different specialization, but otherwise are quite high correlated. So they are active during any working memory task. You have these mid-medial networks that are active during theory of mind, but in generally, when you're following some narrative, so especially when you watch a movie, these areas are actually quite active. Okay, and also involves, of course, on the right side with language functions. And finally, there's this very lateral region out in the tips of Cruz 1 and Cruz 2 that is interestingly associated with autobiographic recall. So one task that activates it is to close your eyes and imagine that you walk from your bedroom to the kitchen of your childhood home. Okay, and that really drives this thing. And it also drives parahippocampal and hippocampals. So we have this new parcellation, this new functional parcellation with functional labels. Now, as you can see, it looks very, very different from the traditional anatomical way to think about different cerebellar subdivisions, right? So here, this is a lobular parcellation that mostly is used in literature, and here is our functional parcellation. Now, of course, they're very different, but which one is better? Is this just arbitrary? Is this just arbitrary ways of cutting up this territory? And we wanted to see this. And if we want to evaluate how good a parcellation is, well, what is the criterion that we can use? And the idea is that a good parcellation should keep similar things together in one parcel and separate things that are different into different parcels. Okay. Now, the problem with this is that when you're close together, when you have two voxels that are very close in the cerebellum, they're of course more similar to each other than two voxels that are very far apart. And that's even the case for the lobular parcellation. Okay, so what we wanted was a criterion where we control this. And so what we're doing here is we're comparing the correlation between pairs of voxels in their function. And we do this by keeping the spatial distance between these voxels constant. Okay, and what we want to see from a good parcellation that for voxel pairs that all have the same spatial distance, like these three voxel pairs, that the voxel pairs that are in different parcels, that they're less correlated with each other than voxel pairs are at the same distance that are within parcels. Now, when we do this actually on an independent task set, as independent from the one that we derived the functional parcellation from, for the lobular parcellation, we see that this is actually not the case. First of all, the correlation between these voxel pairs falls off with the spatial distance of the voxel pair. Okay, so you can see here close by voxels are more similar to each other than far away voxels, but there is hardly any difference whether you are within a lobulus or are you going across lobulus. In other words, two voxels that are actually relatively close together here within across a parcel, so going across parcels, so here, okay, are more highly correlated than two voxels that are within the same parcel but are just a little bit further apart from each other. Okay, so really lobular boundaries do not respect functional boundaries or do not reflect functional boundaries. Now when you look at our parcellation, actually that's not the case. So suddenly you can see, and this is on an independent task set, you can see that actually these boundaries constitute real functional boundaries. So if you step over this boundary, suddenly the correlation in these functional profiles starts dropping quite substantially. Of course, there is some overlap and not all boundaries are equally sharp and we're not fully certain about all boundaries, but it's much, much better and divides it functionally much better than the lobular parcellation. So this means for the cerebellar imaging community, we really should not use lobular boundaries as a proxy for functional boundaries. We really should summarize function in the cerebellum using some different or functional parcellations. Okay, so lobulus is not a good way. So thinking about the cerebellar region as lobuli, 5, 6, cruise 1, cruise 2, as we do often in the literature, is not a good way of thinking about cerebellar localization. And what we really need to think about these more medial to laterally organized functional regions. Now, I'm not saying that our multi-domain task battery parcellation is the be all end all of this. There are many other ways in which this can get more sophisticated, but at least it's much better than the current state of the art. So before I go into the second part, let me just stop here and see if there are any questions.
I have a quick question.
Uh-huh.
Yeah, so the lobules, are they basically demarcated by the cytoarchitecture or by the connectivity to cortex?
Neither.
Neither.
Neither. No, they are just basically, the lobuli are based on the macroanatomical folding that you can see on an MRI scan. So there's a fissure. So there's a big fissure there. I can show you on a, let's see. Let me just, yeah, so here there are these big fissures in there, and this is a lobulus. You can see it, like if you take a brain out and just look at it.
I see. So they're not the equivalent of Brodmann areas of the cortex, which has some psychoarchitectures.
Yeah. So they are, as I said, the cerebellar cortex is very homogeneous across the whole sheet. There are to the naked eye, there's nothing that you can see anything like Brodmann areas. It's completely homogeneous across the whole sheet.
So how about the connectivity then? What pattern does the connectivity give you?
Good. We'll come back to this in the second part. Yeah, it also doesn't respect the lobular boundaries, okay, as you will see. Yeah, that's a good question. Yeah, it goes across, and it's much more medial to lateral organized. There are some differences, and that has to do, I don't know if you know about zebrin staining or aldola C staining. There are these, there's a genetic marker that you can use that shows that there are these zones from medial to lateral that are alternating zebrin positive and zebrin negative, and they do determine a lot of the connectivity as well. But unfortunately, we don't have that map in the human. We know how it looks in the macaque and the marmoset, but not in the human, unfortunately. Great, good. Okay, so let me keep going here. Wow. Whoop. Yes. Yeah. All right. Good. So, so we have our functional regions. Now the question is, to know how these, because the cytoarchitecture is basically the same, the local circuit is really the same across all these different cerebellar regions, at least the first approximation, the function must be determined by which cortical area they talk to. Okay, and so we have some good ideas about which region actually connects to which region, also from resting state data. So there's a paper by Randy Buckner from 2011 who really on 1,000 subject really provides a nice atlas of this. Now the question question we wanted to ask, namely, is how are these areas connected to neocortex? So what we don't know right now is whether there's really this one-to-one connectivity, so this area only connecting to dorsolateral prefrontal cortex, and maybe this area only connecting to, or this area only connecting to frontal eye field, or is there the possibility that there's much more convergence maybe of different cortical areas onto these cerebellar circuits? Now, why is this important? So I think it's very important when you think about function. So if you think about a cerebellar area just connecting and communicating with a single cortical area, you might think that this kind of fine-tunes or provides some learning mechanism for that cortical area. However, if you know that a cerebellar area gets input from multiple cortical regions and then also projects back to the interaction of these regions, okay, then it might actually be enabling some coordination of information exchange between these cortical regions. So your functional theories, your computational theories about the cerebellum will be very different depending on the pattern of this organization. Okay, now we don't know we haven't mapped the connectivity really very carefully here, or the type of connectivity. And so this is a kind of a machine learning project that, again, Maeve King, who's now at Berkeley, and a new graduate student from Iran, actually, Lana Shahani in my lab, have undertaken over the last few years. And we're nearly ready to submit the paper, where we basically use this data set and we parcel the cortex in these different cortical parcels. So here, this is like just an arbitrary parcellation and I think 600 parcels total. And we basically take the data that we have across these different tasks for all these cortical parcels, we take this as our X matrix, and then we try to seek to, with a linear model, try to just simply explain the data in the cerebellum, okay? So this is for each cerebellar voxels, we have the same 29 tasks, and we just try to seek to explain this rich task profile that we have as a linear combination of these cortical task profiles. Okay, and what we can now do is we can generate different types of models that encapsulate different types of theories. So this idea of a one-to-one connectivity, that basically each cerebellar voxel has just one cortical region from which it gets input, would be referring to winner-take-all model. So where for each cerebellar voxel, you just select the best correlated cortical parcels, and that's the one that you declare might give input. Okay, and this is also the model that implicitly Randy Buckner and most other people in resting state actually use. Okay, now the key thing is, like, how can we evaluate this model? And so the evaluation here is that we're going to take the challenge that we're going to give this connectivity model is to take a completely different set of tasks. So this might encode working memory and language tasks. Here we're going to take, movie watching and some reading and some writing tasks. We're going to take a completely different set of tasks. We take the cortical activity pattern only, and we tell the model nothing about the task. We only give it the estimated connectivity weights. And the task of the model is now to predict this entire cerebellar activity pattern in these tasks. Okay, and then we can compare that to the actual measured cerebellar activity patterns. So a good connectivity model, one that really describes the communication between cortex and cerebellum very good, should do this very well, okay, across all tasks. So we have this winner-take-all task, and then we can actually use different linear models that allow for more convergence. So we can do multiple regression, and we can now impose different types of regularization. For example, we can do a L1 or lasso regularization that usually leads to very sparse connectivity models. So basically, for each cerebellar voxel, lasso tries to find a minimal set of cortical regions that are necessary to kind of explain it. Okay. And then you can also, of course, impose L2 regularization or rich regression, which tries to penalize, tries to not have any of these weights particularly large, but tries to distribute them as widely as it can. So this is basically a model where many cortical areas project to each cerebellar voxels. This is where, a small set of cerebellar voxel cortical areas projects to the cerebellar voxel, and this is where one and only one cortical area projects to each cerebellar voxel. So now we can compare these different models, and we evaluate it by just simply correlating across these tasks, correlating the predicted activity patterns with the actual observed activity patterns in those subjects. And we do this, the fitting within subject and also the evaluation within subject. Now, So we have these three different models. We also don't know what is a good resolution basically to construct the features here of the cortex. What is a good resolution to do this in? So we varied this from about 80 regions for the entire cortex to about 2,000 regions for the entire cortex. All these parcellations are completely arbitrary, and I'll come back to that. So this is just an arbitrary grid that we're going to put across the neocortex. Now, when we look at the predictive accuracy as the function of the fineness of the cortical parcellations, you see that for our data, it doesn't actually matter very much how many cortical parcels we take. Rich and Lasso actually perform relatively similarly. Lasso performs slightly less good. There is actually significant differences back here. Rich actually performs best. Now, interestingly, the winner-take-all consistently performs lower than the other two methods. So the winner-take-all really is significantly lower than the rich, basically for any possible parcellation. Now, one disadvantage the winner-take-all has is that it relies, of course, only on one single cortical parcel. And that's a concern because we measure the cortical activity also with noise, especially when the parcellation gets smaller. Right? This noise level will increase. So we need to account for this. And we also need to account for the measurement noise we have on the cerebellum. So we want to ask how good this 0.2 predictive accuracy sounds very low, but you need to keep in mind that this is unsmooth, single subject, single voxel data. Okay? So what we can do is construct a so-called noise ceiling. We can ask the question, if we had the perfect connectivity model, if we knew the true connectivity weights, what would our performance be? Okay, and that's the noise ceiling, and then we can normalize our predictive performance based on this noise ceiling. And what we see here is that, again, Rich wins over Lasso, wins over winner-take-all, but that we are explaining about or predicting about 60% of the explainable or consistent variance of these activity patterns. So it's actually a quite powerful model. Right. So of course, you might argue, well, this is basically for the cortex cut up arbitrarily. There are many functional parcellations that are actually motivated. So here's one, for example, motivated by resting state data or derived from resting state data. These are seven regions. And if you do that using that cortical parcellations, you get actually worse predictive performance here in the three models. But again, the winner-take-all is substantially worse than the Lasso. So what we conclude from this is that the functional profiles in the cerebellar voxels are actually best predicted by a combination of a few in Lasso or many, like in Rich, cortical areas. So the question is, does this hold true over the entire cortex or entire cerebellum, or are there differences? So what we're looking here is now the connectivity weights for each of the cerebellar regions that we had established in the first part of the talk. And we just look at which areas of the neocortex project to this, or like would be connected in the connectivity model to these areas. And so basically what we find here for the left-hand area, we of course find right motor cortex and sensory cortex. So very focal. For the right hand, it's left motor cortex and left sensory cortex, as you would expect. For the saccade, for the oculomotor vermis, we see that there's input coming from the frontal eye field, intraparietal sulcus, and extrastriate areas, as you would expect. So this is a little bit more distributed. This is very focal. This has more distribution here. Right? Action observation is this network of this action observation. It looks a little bit fuzzy here. You see for the working memory network, left and right, you see this very clear dorsolateral prefrontal and parietal areas projecting in a lateralized fashion. For these mid-medial networks, you get this, the temporal, inferior parietal, and middle and inferior frontal networks, again lateralized to the respective side. And then finally, for autobiotic recall, this looks a little bit messy, but you can take my word for it that there's connectivity to the medial side and also to the frontal pole of the neocortex. So when you look at these patterns, one striking thing is that the motor networks are actually very focal. So they have this small area, very focal area that seems to project to them, whereas other places like the language network or the working memory network seem to need a number of different regions to actually be explained. And so for this, we use the lasso model that tries to minimize the number of regions that it needs to explain it, and look at the area, the percentage of cortical surface that is marked by this. And what you see in individual subjects, in individual voxels, what you see is that the motor areas 1 and 2, the left and right-hand networks, indeed have a lower area that is necessary than most of the cognitive networks that are depicted here. You also can see here, this also holds true when we do this voxel-wise. You can see here, this is this analysis projected on the cerebellar surface. You see anterior and inferior cerebellum need very little cortical input, or very focal cortical input, and these need to actually more areas to explain their activity patterns. The same you can derive by using a spherical dispersion. So this measured not how big the area is, but from how many different parts of the neocortex it comes. So this is much more focal and this is much more dispersed here. And you can see that again, the difference between the motor regions and the more cognitive region. So this suggests that this one-to-one connectivity is probably not the right way of thinking about the cerebellar circuit, but that maybe for some areas like the motor cortex, motor circuits in M1 and the hand areas of the cerebellum, there is really this one-to-one connectivity with sensory motor regions, but that many regions like these working memory circuits in the cerebellum not only get input from the dorsolateral prefrontal cortex, but also from parietal and temporal areas at the same time. Now, this convergence might really suggest that there is really different functions, that really here, how this cerebellar region works with this single cortical region might be structurally different from how these areas work with this group of cortical regions. So maybe these ones coordinate activity much more, whereas this one fine-tunes local activity more. Okay, I'll take a little break again, just to see if there are any questions up to this point.
Can I have a criticism, Jorn?
Yes, absolutely.
Okay, so all you've shown is that one region of the cerebellum, one of those functional regions, have correlation with multiple regions. Let's say for the vermis, you have FEF, IPS, and some other. But that doesn't. But that's just correlation, right? So it's so if it's a graph, all of these regions are going to be correlated with each other.
Yes.
So I guess one needs to do either. If you just want to do correlation, one needs to either do partial correlation to see if maybe one of these.
Yeah, it's not. So I just want to clarify. So yeah, your point is very, very good. So first of all, clearly it's all based on activity correlations, and so we can't infer direct connectivity from it. Now I want to clarify that the maps I showed you aren't just correlation maps. So it's not what people have done in resting state data. This is multiple regression models where it's basically partial correlation, as you say. Okay. So all the regressors are entered at the same time into the same model. And then the regularization, for example, lasso now tries to because it's you have more regressors than you actually have observations. You need to regularize, and the form of the regularization, lasso versus ridge, now gives you a certain a level of sparseness of this input. Like lasso, with high regularization, tries to really find one or two necessary regions and try to set most of the other ways to zero.
But you haven't done stepwise regression, so you put all the X's.
You can do that. You can do that, and we've done this.
Right.
Yeah.
Right. So if there is correlation between the X's, between the independent variables, then if you put them all into the regression model, the model will find some of the best beta to explain. So all I'm saying, maybe like a more strict analysis requires either a stepwise kind of regression to put one by one to make sure that the residual of each region still can be explained, can be picked up by the other.
So Ali, so stepwise regression. So we have done stepwise regression. And it gives. I mean, it is lasso basically is you can think about it as an automated version of stepwise regression.
Right,
stepwise regression is a little bit is not the best statistical technique because obviously it's not going to find the best sparse method right now. So we also have done a very careful simulation. So I was very critical of this approach in the beginning when we considered this, and we've done a lot of simulations where we actually generate data using the correlation structure that we have in the cortex with all the codependencies between these different regions and simulate data under different connectivity models and see if we can retrieve it. Can we retrieve the true model using this thing? And that's the case.
I see.
So the correlation of the cortex, and this was one of the key design criteria for our multitask battery. So basically what we wanted was a battery of tasks, of 60 or so tasks, that tries to, when you look at each of these cortical regions, make them as independent as you can. Okay. Normally, if you only have 3 or 4 tasks, you have all these very strong correlations between them. Yeah. But when you do this very broad task set, you get actually start dissociating these cortical regions.
Okay.
Okay?
So
it's not perfect, but
right.
it's quite good.
One clarifying question. So in the beginning, you said that Peter Strick had this connectivity analysis. Probably other anatomists have done. So what does that say? Does that say that one of these regions receive input from multiple segregated regions, or no? Like
it's a very good question. We don't know from this. So right, he injects in anterograde, and in this paper, in the Kelly and Strick paper, and this is the only data that's out there so far, is that he injects retrograde and anterograde viral tracer into a cortical area. Okay. So what he gets is the cerebellar areas, and then disynaptic tracing down to the cerebellum. And what he gets is so basically what he gets is the areas in the cerebellum that project to this area, and the cerebellar areas that region projects to. What you don't know from this is whether other cortical areas also project to that same cerebellar region.
So he hasn't done overlap between the two?
That is very hard technically. I mean, you can talk to him. I'm not an expert on this. Yeah, it's very, very hard to do multiple. I think now with optogenetic techniques, I need to talk with Adam Handman, who does some of this work, whether it's now possible. I mean, Adam has done with multiple tracers, optogenetic tracers, in the same animal. So it's also, in Peter Strick's work, is also for the retrograde and the anterograde is done in separate animals. So so you have the intersubject variability in there and so on. So it's quite tricky. So yeah, that data and the restings data so far doesn't address this problem of convergence.
I see.
Even though people talk about it as one-to-one connectivity, and certainly if you read Peter Strick's paper, you get the impression then that's his favorite kind of mental model of how they're connected. But actually the data, and he admits this in the paper as well, you can't exclude the fact that there might be more convergence than one-to-one.
Okay, thank you.
Okay, great, good. Okay, excellent. Okay, so I guess I have 10 minutes left. So just the last thing, this is just the first steps. In trying to see if we can use functional imaging to something more than just what we have so far. So far, you use functional imaging to really look at the cerebellum and find the different functional regions of the cerebellum, okay, and know which task activates these different regions and how they are connected, which cortical areas they're connected to, and how they are connected, maybe. So that's already a lot. But what we can't say so far from this data is what the function of each of these cerebellar regions is in the context of the cortical region. Okay? So that's really what we want to know. Like, what does the cerebellar circuit do in this motor cortical circuit, and what does the cerebellar do in this working memory circuit? And we have no answer to that so far, because basically we just observe activity here and activity there, but that doesn't tell us what the function is. And one of the main limitations is, of course, of the cerebellar fMRI is that we're mostly looking at the cortical input. Now, let me turn this question, whether we can actually learn anything from fMRI, around and argue that maybe we can't. Okay, so imagine we're going to go now for 2 years away and analyze many big imaging data sets, which we are doing, and we come back in 2 years and we have a connectivity model that not only explains 60% of the noise ceiling, but 100% of the noise ceiling. So for any task you give me, I just need the cortical activity pattern, and I can perfectly predict the cerebellar activity pattern up to the measurement noise level. Okay. Just imagine we had a connectivity model like this. Now, that would mean that I can retire, because now there is no cerebellar imaging that really needs to be done anymore, because we just have to record the cortical activity, and then we predict the cerebellar activity. There's nothing that you can learn anymore from cerebellar fMRI. Basically, the only conclusion we can do is, yes, this cerebellar region works together with this region on the function that this cortical cerebellar column is involved in. Okay? You can't really ask what does the cerebellum specifically do in this. Now, the fact is that even with the best models we have right now, we are predicting, and this is on completely new data, completely new subjects, new scanner, and so on. We are predicting about 60% of the explainable variance. This is normalized to the noise here. Okay, that also means that there are 40% that we're not predicting. That is unexplained by this best static connectivity model that we have. Okay, and this gap, this 40% gap, I would argue, is something that we should look at. That is actually our chance to learn something specifically about cerebellar function. And so Lana and I, in her first project in her PhD, wanted to look at this in an area where we actually know the answer. And the circuit for which we have a pretty good idea what the role of the cortex and what is the role of the cerebellum is the hand area of the human cerebellum. So this is the hand area for the right hand connected to the left primary motor cortex. We also know what the cortical areas are, and they're working together to give rise to hand movements. Now, from patient data, we know that if you have a stroke in this cerebellar region, you develop ataxia, where one of the main symptoms is dysdiadochokinesia. Dysdiadochokinesis, which means it's the inability to produce rapid alternating movements with fingers. Okay, so they have problems in really tapping very fast, alternating with the fingers. Now, another interesting thing is that these cerebellar patients have no problem generating force. So when you have somebody like this in a clinic, a degeneration or cerebellar stroke patients, they can squeeze your hand fully. They have very little force impairment. On the other hand, if you have a stroke in primary motor cortex, well, you're going to have problems in rapid alternating movement, but you're also going to have a lot of problems in force generation. So it's often weakness allowed to it. Now, okay, so imagine we record the fMRI activity in both of these circuits at the same time, which we can do. What would we predict? So if you tap with low force and very slow, you get some cortical activity in this region here on the x-axis, and you're also going to find some cerebellar activity on this region here on the y-axis. Okay. Now, when you increase force, you're going to increase cortical activity, and because they're connected, you're also going to increase cerebellar activity. So far, we haven't really learned anything. Okay. Now, if it's a fact that when we increase speed, we get some increase in cortical activity and the predicted increase in cerebellar activity, well, then the static connectivity model really works, and we can't really learn anything from it. But based on the clinical data, if there's some gating of this cortical input to the cerebellar regions, meaning the cerebellum becomes especially active when it's actually needed to do this, we would predict that the cerebellar activity is now higher in respect to the comparable cortical activity. Okay? So that's the logic of this idea. So it's very important to look, basically you see increases in force, increases in cerebral activity, both for force and for speed. And to compare them, you really need to look at it in relationship to the cortical activity. Otherwise, you can't make any inferences from fMRI about specific cerebellar function. So this is what Lana did. So she has just a very simple experiment. 5 conditions. Force is either 2.5 newtons, 6 newtons, or 10 newtons. 10 newtons is nearly as hard as you can press. And you go at 1 hertz, so 6 presses over 6 seconds, up to 10 presses for 6 seconds, and 18 presses for 6 seconds. This is basically maximal speed here. So cerebellar patients would have trouble doing this here. Now, when you look at the neocortex, if you look low force to high force, you get this nice activity in M1 and S1 as expected. Also, when you increase speed, you get very similar activity increases in contralateral motor cortex and sensory cortex. Okay? Now, when you look at the cerebellum, you get, with forces, as predicted, you get increases in the right hand areas, so anterior and posterior hand areas. You get also increases with speed. Now, here you can already see that these increases are bigger than the ones for the force, as we might have predicted with the functional involvement of the cerebellum for coordination of rapid movements. Now, we need to do this formally, of course, so we used here in this analysis just this hand area RI from our task and plot that on the y-axis, and we're going to take the we're going to take the subject-specific cortical activity and multiply that by the weights of our cortical connectivity model. And we can use here we use the rich because it's the most conservative one. You can see here that there is input, that rich says there is input from these areas that are also activated here during the speed task, and we plot that predicted activity on the x-axis. What you see here with speed is that there's this nice linear relationship. So this is low force, medium force, and high force. As force increases, so does the cortical activity, and linearly the cerebral activity. So far our static connectivity model holds. Now when we look at the speed condition, so the medium speed is a little bit lower activity than the medium force on average. The high speed has a little bit higher activity than the high force. That's not so important, but important is whether these 5 dots all lie on a line or not. And when we fit a line and then look at the residuals, we can actually see that the differences from speed and force here are actually, that the residuals are actually significantly different. So this is always below the line and this is always above the predicted line. We can also do this on a mapwise approach where we do this regression analysis basically for each voxel of the cerebellum, and we see that these areas that are more activated for the speed condition than the force condition, as compared to the difference in the neocortex, as compared to the areas in the cortex, right? We see that we find the right-hand area, anterior and posterior. When we turn this contrast around and look for areas that are more activated in the force than in the speed, relative to what you would expect from the neocortex, we don't find any region. So there's no region that cares about force in the cerebellum. All right, so this is somewhat encouraging. So we started from a known functional involvement of the cerebellum and predicted that we should see actually some modulation of the cerebellar input for the similar cortical activity patterns or similar cortical activity level, right? For the speed task, the cerebellum is just more activated than for the force task, relatively to what the cortex is doing. So how is this possible? Now, there are multiple areas where this could occur. So first of all, so there must be something in the connectivity that is actually a dynamic, that depending on task, either the cortex tells the cerebellum a lot or not, or provides a lot of input to the cerebellum or not. So there are many things that could be this, and unfortunately with fMRI we can't tell. It could be that some tasks specifically activate pontine projecting neurons in the neocortex. The pontine nuclei are, of course, it's not just a relay station. It's not just there's a synapse and it goes out. There's actually some information integration and gating going on in the pontine nuclei. And this might actually decide where, depending on the exact what's going on, the process of the cortex might decide that it really needs cerebellar help or not. Okay, it might also be internal dynamics in the granular cell layer. So there's the internal dynamics that could give rise to this. Okay, so what we've seen then is that we really, with this approach, we're hoping now to go not only in the motor circuit where we kind of know the answer, but also go in the cognitive circuits and with the established connectivity model, figure out what these areas specifically are doing over and above the fact that they're activated during the task. When you compare it to the corresponding cortical regions, which task features do activate cerebral circuits especially. Okay, and that is one thing that we can do in fMRI with human that would be very hard with any other methods. And from there, looking across regions, we hope that we can identify the different processes in the different tasks that really call on specific cerebellar function and maybe put together or come closer to an answer what the cerebellum in general is doing to help the cortex support mental function. So I want to just acknowledge all the people that are involved. So there's, of course, Maeve King and Carlos Hernandez, who've done a lot of the work in this project. Ladan Shashahani, who joined the lab 3 years ago, who has worked in this project, Chi for the viewer, Madhusri and Raspoldrag I mentioned, and of course Richard Ivory, who has been involved in much of the work. And I thank you for your attention.
Okay, thank you so much for the great talk, and very energetic and very informative. If there's any questions from the audience, there are some questions in the text box, Jorn. You may want to take a look.
Okay, so high resolution image of the cerebrum were obtained with fMRI 70. Yeah, so the data that I showed was mostly from the 3 Tesla scanner because we want to do whole brain. So this is actually relatively low resolution, about 2.5 millimeters. We are doing now high-res imaging in 7T to look at deep structures, pontine deep nuclei, and that's being done at 1.2 or 1.5 millimeter resolution. Yeah. And then how can we interpret your findings with respect to the much higher density of neurons in the cerebellum? Maybe that's the, you're asking here, maybe is that the reason for the diverse connectivity to the neural cortex? That is a very good question. So what we did here in the connectivity model is that we went, we used, so it's right that even with a small voxel size that we have, we're going to mix across different areas. And so when you build a connectivity model, it can be that some of the convergence is just partial volume. You have some, you mix across some cerebellar areas, some of which gets input from this area, and this they get input from that area, and maybe they just sum together. So that is possible. We tried to do this carefully in that we did this regression voxel-wise. So we did it for each individual voxel in each individual subject. And that's very important because if you average across subjects and you average across voxels in a region, you're definitely going to be problematic. So all the data that I showed you is this voxel-wise regression. Now, it is true that voxels themselves average some signals, although, at this level, I think probably the function, now that you're like in this 1 millimeter area, you're probably pretty homogeneous in the function. Although we can't exclude that that's the case. So that's a very good question. Good. Other questions?
So you can raise your hand or you can type it in.
Yeah, speak up.
Right.
Don't be shy.
And so, Jorn, at 7 p.m. local time here, which is 1 minute,
Okay.
we can all move to the panel address that I just typed in. And for the people who want to have more in-depth discussion with you.
Okay, that's great.